Now we have our last talk of this event. This is by Dr. Pamela Kunz. She is an esteemed leader in the field of neuroendocrine tumors. She is a medical oncologist at Smilow and Yale Cancer Center. She will be talking about clinical trials, the latest and greatest, giving us a dive into the future perspectives. And Dr. Kunz's fun fact is that she's a mom of 3 boys and spent most of her outside work time on a baseball field or a basketball court. Dr. Kunz.
Good afternoon. It's really a pleasure to be here. Thank you so much for staying till the very end. So I'm going to be talking about clinical trials, how to interpret them, how to find them, why they matter. Okay. All right. Moving right along. So why are clinical trials important? Well, they are a key research tool for helping us advance knowledge. Clinical research is done to learn whether a new approach is safe, whether it's effective, and then which treatment strategies work best for certain groups or cancers. And clinical trials, I think, really provide hope. I know at the end of many of my clinic visits, I'm able to tell patients that there's this new drug, cabozantinib, that just got FDA approved this week, or that we have a new trial that's starting, and I think it's incredibly hopeful. Yet only 7% of US adults with cancer participate in clinical trials. So let's talk about some myths and facts. So the myths are listed here on the left, and the facts are on the right. So clinical trial participants are, quote, guinea pigs. That's a myth. So the fact is that there are actually strict guidelines in place to inform clinical trial volunteers and protect their safety, so you will know exactly what you're getting into if you choose to participate in a trial. You'll know whether it's a randomized trial. You'll know if there's a placebo. There's a whole process called the informed consent process. If I join a clinical trial, I will receive a, quote, sugar pill or placebo instead of real medicine. The fact is that that can be true, but you will know placebos are generally only ethical if no standard treatment is available. And I actually think that we probably will no longer have neuroendocrine tumor trials that involve placebos, because we now actually have so many available therapies, which is very different than even 10 years ago when we didn't, in which case placebos were actually ethical. Once I decide to participate in a trial, I can't change my mind. Well, that's not true. What is best for you trumps anything that's best for the trial. So if you ultimately decide you want to come off of a trial or it's no longer for you, you can make that decision. There are no costs associated with a clinical trial. So many aspects of a trial are, in fact, covered by the clinical trial sponsors, but some participants may have copays for standard items. So for example, if you're standardly getting clinic visits, those may go through your regular insurance and you may have a copay. Or a scan, maybe a standard of care scan, and that may still go through your regular insurance. Clinical trials are only for patients without other options. That's also not true. So there are clinical trials available for all stages. In fact, we have an open trial right now for patients with high-risk pancreatic neuroendocrine tumors following resection. Half the patients get capecitabine temozolomide, and the other half get placebo, or I should say best supportive care, because that's the standard right now. Okay, talking a little bit about the path to drug approval. So to get a drug from the lab in test tubes and testing in mice to FDA approval can take about a decade. We hope to shorten that process, and there are some efforts in doing so, but all drugs have to go through the preclinical to phase 1. So phase 1 tests safety. If it passes phase 1, then it moves to phase 2 or phase 3. And phase 2 and 3 trials are testing hundreds and perhaps even thousands of patients in more common cancers. After drugs go through phase 2 and 3, a drug then gets submitted to the FDA, and that's a process in and of itself, and that's actually what just happened this week with cabozantinib. And then after FDA review, we're still collecting data on safety, and so that's called phase 4. So that's real-world data that we're still learning. So I wanted to just briefly touch on sponsors for trials because I think that's also something that's not well understood. So a sponsor means who is responsible for the conduct of the trial. There are three primary sponsors, something called investigator-initiated. That might be me coming up with a clinical trial idea at my institution. I'm going to start a trial. It's maybe a smaller study. An industry-initiated study is usually a larger study. The sponsor is that pharmaceutical company. They can do phase 1, 2, or 3 clinical trials. And then there are national clinical trial network trials. Those are sponsored by our federal government. We actually have a number of federally funded clinical trials for neuroendocrine tumors that are often the really practical questions, ones that may not be done by pharmaceutical companies. So federally funded NCTN trials represent a really important way to ask these practical real-world questions. I'll give you some examples here. These are 5 that are listed for NETs. So the first one, lutetium dotatate versus CAPTEM in pancreatic NETs, lutetium dotatate versus everolimus in lung NETs. So Alliance and SWOG are the names of the National Clinical Trial Network groups. CAPTEM versus observation after surgery is the one I just mentioned. Lutetium dotatate retreatment. And then temozolomide versus tem and olaparib in paraganglioma and FIO. However, that first one just got closed due to low accrual just a few weeks ago. And we risk other important NCTN trials closing if they don't accrue. And I think what I've heard from some patients, and Lisa and I were talking prior to this, I think all of these drugs that you see listed here are available as standard of care. So some patients are saying, oh, well, my doctor is going to give this to me regardless of whether I go on the trial. That's possible. But I think there's something that's very important about paying it forward and about contributing to research that will actually help answer critical questions. Ludotatate versus CAPTEM. There's equipoise. It's a coin flip. We do not know if one is better to start with than another. We actually will now never know the answer to that question because that trial has just closed. So design and interpretation of trials, we have eligibility checklists. So there are very specific criteria that are involved in being eligible for a trial. So which primary site, pancreas, lung, small bowel, which grade or differentiation, what stage, is the cancer progressing or not, is prior treatment allowed or not. And then we have endpoints that in the kind of clinical trial speak mean how do we define success of that trial. One that's very common in NETs is called progression-free survival. So that means how long does it take for the tumor to grow, or overall survival, how long do patients live, response rate, how much does the tumor shrink, and then safety, another very important component. I'm going to walk you through how to interpret. These are kind of called Kaplan-Meier curves, and they're ones that as a researcher we look at to help define successive trials, but they're kind of hard to know how to look at them. So this, I think there is a pointer, good. So the horizontal axis is usually time. Oh, I think this is like patting your head, patting your stomach, and whatever the expression is. I can't do both at once, let's see. So the vertical axis is progression-free survival. So that's here. And then this dashed line is something called the median progression-free survival. So it's kind of like the midpoint in terms of where patients fall in terms of whether they're progressing or not. And then the hazard ratio means the hazard of progression or the risk of progression. A low number is good for both the hazard ratio and the p-value. And then we want to see separation of the curves. So this is an example from a clinical trial that I led of CAPTEM versus temozolomide in patients with advanced pancreatic NETs. What we're seeing here is that the yellow line was the combination line of capecitabine temozolomide compared to temozolomide alone, so that the fact that that line is on the top and has a longer median progression-free survival meant that that group of patients did better. They had a longer time before their cancer progressed. And so that meant that for this clinical trial, we know that CAPTEM actually was better. So the other thing we measure is how much the tumor shrinks. That's called response or response rate. We determine this through some criteria that have an acronym called RECIST. It stands for Response Evaluation Criteria in Solid Tumors. And a lot of patients will ask, well, why don't you measure all of the tumors? Well, in a clinical trial, we don't. We take a maximum of 5 representative tumor spots and a max of 2 per organ. And then we measure those really precisely over time. We actually have a team. It's often the same radiologist that's reviewing those at different time points. I'm going to go into this in a little bit more detail with a cartoon on the next slide. So imagine, this is a really bad drawing of a liver, by the way. So this is a liver, and it's in the 2 dimensions. Imagine there are 2 liver lesions here. I've just made a table here. So liver lesion 1 is 2.5 centimeters. Liver lesion 2 is 6.5. We take the sum of those, and then at the next time point, after 3 months, we do those exact same measurements again. And this particular patient, this fictitious patient, has had some shrinkage. So 2 centimeters, 5.5, the sum, 7.5, and there's been 17% shrinkage, so that's called stable disease. And then after 6 months, we measure it again, and there's been 44% shrinkage. By hitting the 30% shrinkage mark, that is determined, that equals partial response in those criteria. So this is what goes on behind the scenes when someone's on a clinical trial. All these very quantitative measurements are made in order, that's one of our determinations of success. So then we put it into a table, and as mentioned here, so partial response is 30% or more shrinkage. Stable disease is anywhere between 30% shrinkage and 20% growth. And progression of disease is 20% growth. And so this particular clinical trial, you can see that CAP-10 arm had 40% shrinkage or response rate compared to temozolomide of 34%. So the combination arm still did a little bit better. The third key endpoint is safety, really critically important, although these particular agents had been studied in earlier phase trials, so we really knew the safety, but we generally always assess that, even in phase 2 and 3 clinical trials. This is also graded very quantitatively. We look at the severity, and that's on a scale of 1 to 5. We look at whether it's related or not to the study agents. And then we sometimes are now using patient-reported outcomes. We have there's actually like an online app and a tool to help us with the grading, but this is what goes into the reporting. So I'm just going to end with a few slides on like hot trials to watch. So for neuroendocrine tumors and pheoparaganglioma, systemic treatment, thinking about cytotoxic therapy, there are 3 trials, and this will mostly serve as a reference. I'm not going to go through all of these in detail, but the SWOG study is the study of giving CAP-10 versus observation after surgery. The Sorrento trial is a relatively new trial that's octreotide depot every 2 weeks versus investigator's choice. This is an outstanding question of at time of progression, can we go up on octreotide or lanreotide if patients are already on a standard monthly dose? And then this is a trial for metastatic paraganglioma and pheo of temozolomide versus 10 plus olaparib. And then neuroendocrine carcinomas, we have a large study looking at platinum etoposide plus or minus immunotherapy. And then lots of radioligand trials. So again, the key takeaways from this is that we're starting to look at lutetium dotatate and similar agents compared to other treatments. That's really exciting. We're, again, moving away from comparison with octreotide. So there's one of lutetium dotatate versus everolimus. Net retreat, can we repeat ludotatate at time of progression? Actinium-225 is an alpha therapy, so that's a new radioisotope trying to improve on lutetium dotatate. So alpha therapy, and you probably heard about this recently from Dr. Thompson, but alpha therapy theoretically travels a shorter distance and has a higher linear energy, so theoretically more effective and less toxic. And then there's a new study that will hopefully open this spring of looking at an oral somatostatin analog to control carcinoid syndrome. I get asked frequently about why can't we use immunotherapy in neuroendocrine tumors? And why can't we apply some of these kind of other novel approaches that we're seeing success in other agents like pancreas cancer? I think we're not quite there. It doesn't mean we're not going to get there. Immunotherapy in neuroendocrine tumors has generally not proven effective, but there may be some new ways we can think about doing it. Even just this week, there was an early publication from something called CAR-T in neuroendocrine tumors. That's chimeric antigen receptor T cell therapy. It's a type of immunotherapy that is in a clinical trial. So stay tuned. I think we're really still hoping to capitalize on some of the successes in other cancers, but it's not proven effective yet in NETs. So how can you learn about trials? There are lots of ways. So number one, ask your treating physician, and my time's up, but I have only, I think, one slide left. So ask your treating NET specialist. Go to your regional cancer websites. Go to clinicaltrials.gov. Go to patient support groups, and there are many, including NCF, NANETS, NetRF. And then finally, so hopefully to leave you guys with some hope, we've made many advances in the last 10 years, all thanks to clinical trials, really. And clinical trials are critical to making progress, incredible reason for hope. And we have a lot of novel agents in the pipeline. And a shout out for any of you who've been a clinical trial participant, we thank you. And if you meet future clinical trial participants, please thank them also. So thank you so much, and happy to take questions.